Cremolina is one of the worst terrestrial weeds in South Africa and across the world. But in South Africa, we have for a very long time been trying to find out where our Cremolina odorata comes from. And we haven't been able to answer this question for many years. And recently we've got some really strong evidence that we now know the origin of our South African type of Cremolina odorata. Obviously I can't tell you that until the end of the talk. And that's the reason why Costas decided on this title, that there's now strong evidence that puts a very old question that Costas in particular has been grappling with for many, many years. So in green there we have the native range of Cremolina odorata. What you'll notice is that it's a very, very large native range. It's a very large native range. And then also the introduced native range, the introduced range is also very large. And you can see that there are three distinct groups. There's a West African invasive population, there's this Asian population, and then we've got quite an isolated Southern African population. I must just stress that this map has expanded. This data is from 1992, and today there will be many more dots, especially in this area. So within South Africa, this plant is very homogeneous in appearance throughout the range. So all of our plants look the same, which is typical of many invasive plant species. But then also across other parts of the introduced range, so that's in West Africa and in the whole of Asia, it's also very homogeneous, but it looks different. There are morphological differences between the two types. So we refer to the one as the South African biotype, and we refer to the other as the Asian West African biotype. And the aim of this study was to find out where our South African plants came from. So where in the native range did our South African plants originate? But why is this important? And many of us know that biocontrol agents can be very host-specific, and they can be host-specific within genotypes of the plant. In the case of Cremolina odorata, it became very apparent while working on the plant that this was definitely an uncertainty in terms of biocontrol of Cremolina odorata. To illustrate that, this is a biological control agent for Cremolina odorata. We don't have it in South Africa. It's called Cecidocarus connexa. And it forms these very damaging galls on the stems of the plant. And this fly species is so host-specific that many years ago Rachel McFadden did some work on it. And so this is the top of South America. Can you all see that? It's not a very big map. No, okay, well, on this side of Panama, she collected plants with galls on it, and she took them, it was about 400 kilometers down the road, and popped them on another genotype of the plant, and this insect could not accept that different genotype. So it's so host-specific that it won't accept plants from just a couple of 100 kilometers away. So when we collect biocontrol agents to control Cremolina odorata, we are going to have to get biocontrol agents from the right place, otherwise they might not accept the weed. And if they do accept it as a host plant, they might not do the best job that they should do. So the early efforts in finding out where Cremolina odorata, where our South African biotype originated from, the first place to look was in Herbarium Specimens. And this was done in Kew and at the Smithsonian in America, in USA, sorry Jackie, in the USA. And they found that morphologically, our South African biotypes were most similar to the Jamaican and Cuban specimens. But then there was a very nasty specimen that stuck out from Guyana, and there was only one of them. And then later, Stefan Neerse did a field trip for another reason to Jamaica, and he came back with a live plant that looked exactly like our South African biotype. So that gave people an idea that our biotype may have come from Jamaica. So this is where Costas was trying to find the origin of this plant. He thought it could come from Cuba and Jamaica because our herbarium specimens looked really similar, and then there was that one single herbarium specimen from Guyana. So the next step to try and confirm this was to look at some genetics, and using the ITS region and 54 different Cremolina odorata samples. The ITS region is just quite a variable region of DNA, and it was sequenced. And these are the big trees that were produced from it. And if you like these sort of trees, you'll notice that there's only very high bootstrap values. And these bootstrap values are basically a measure of how real the different groups are. And it's important to note that all the high bootstrap values are really, most of them, are all at the bottom of this tree. And that's because these are all outgroup species. They're not actually Cremolina odorata. So what we've got here is poorly supported groups, but we do have a lot of South African plants. These, you'll have to just take my word for it. These are plants that say SA, and they're mixed up with Jamaican plants, and they fall into a group, but they're not a strongly supported group. But we also have South African plants that stuck out with other plants, and Jamaican plants that stuck out in other parts of the tree. So we didn't get much resolution from this technique. Then a bit more non-molecular work was done, and there were various field trips done all over the native range of Cremolina. And they found that Cremolina odorata that was identical to our South African Cremolina odorata morphologically, and others that were very similar to the South African form, were found in Jamaica, Cuba, Puerto Rico, and there's a record of it from the Bahamas. But it's also important to note that lots of other types of Cremolina were found in these places. And it's very, very variable across its native range. Another bit of evidence that came was from looking at McGibbon's catalogue of plants that were brought into Cape Town in 1858. This was a botanical gardens, it wasn't Kirstenbosch Botanical Gardens, but he had a full record of all the plants that were brought in, and they said that they brought Cremolina odorata in, and that they brought it in from Jamaica. Kostas Zacharidis has spent a lot of time looking at these plants. He quickly ran through this presentation with me before I've given it, and he told me that this is a photo of the plants in Jamaica. This is the South African biotype, and this is the typical native form. And he said it's very clear that you can see the difference. I think it just shows you that Kostas has really, really looked at this plant. Okay. But most of this work until now was anecdotal. It really looks like it's coming from Jamaica or Cuba, but there was no firm proof. So what we did was we used a higher resolution genetic fingerprinting technique called the ISSRs. It really doesn't matter what that is, but it manages to look at a more fine scale, at finer scale relationships than the ITS region. We had 39 samples, 27 from the native range, getting as much of a spread as possible, and we got 7 from South Africa and 4 from the Asian West African type. Now, some of those numbers are pretty low, but given the very small geographic range of the South African biotype, 7 did give a good cover of the area. For the Asian West African biotype, has a huge introduced range, and we were never going to get an answer as to where the Asian West African biotype comes with only 4 samples. And this is the data. Last time, all of the last tree you saw, all of the high bootstrap values were down towards the end of the tree. In this case, we have much nicer, higher values at higher clades, at more detailed clades. And overall, here we've got plants in Group A. We've got all of the South African plants, all 7 of the South African samples, and they mix in with Jamaican and Cuban samples. So Group A is only Jamaican samples, Cuban samples, and all of these South African samples. Then there's another sister clade here that has 2 other Jamaican samples. What that tells us is that our Cremolina came from Jamaica or Cuba area, and all of our South African samples are in there, so that's the one place that it did come from. And then outside of that group, we've got 2 Jamaican samples. That's because there's lots of plants in Jamaica that didn't lead to the plants in South Africa. In Group B, we had all of our Asian West African biotype samples, and they mixed in with samples from Trinidad, Guyana, and Venezuela. So we don't have very good resolution as to where the Asian West African biotype came from, but we weren't expecting to find any. And then Group C, D, and E are from all over Brazil, Argentina, Mexico, and all the other parts of the native range from where the South African samples did not come. So the main findings and implications are that the South African biotype of Cremolina came from Jamaica or Cuba, and it forms a very distinct group. It's also important to note that the genetic variability within South Africa is very low, and that means that if we choose the right biocontrol agent that is well matched to our genotype, we could get very good control, because low genetic diversity is a very good thing when controlling a weed. It means that that's where we need to go to collect our biological control agents. As I said, we didn't find out whether the Asian West African biotype originated from, but at the moment we've sent all of the samples that we had off to China, and there are people working on it there on a very large-scale study to find out where that biotype came from. Another thing that it showed us was that the Asian West African biotype and the South African biotype did come from different places. And we expected that from the morphology, but it's really nice to have it confirmed through the genetics. Thank you very much.